I just arrived this morning, and I'm still on the Eastern time, so I was thinking we were running an hour late. I was telling Gökhan, what are we going to do? And he reminded me that we were like an hour early. But I was eliminating some of the slides, so I have only 100 slides left, so I'll try to go quick about that. But what I will try to convince you today is that the introduction of the next generation genomic technologies and how these are becoming of daily use and how these are influencing our decisions in medicine day by day. And I can guarantee you that if not all of our genome being sequenced during our lifetimes, for our children, their genome definitely be sequenced, and medical decisions are going to be made based on those. We're still at the sort of early stages of understanding that. But if you think about why we use genetics, it really is a very powerful mechanism, because this classic scientific approach, it relies on us being smart and generating hypotheses and then testing those hypotheses. So when I arrived from Turkey in 1990, I had no choice but to find another approach of not being able to produce hypotheses. How do you generate hypotheses? Genetic research gives you that opportunity, because you take a genome-wide, unbiased approach. You take the data, you analyze the data, and the data shows you. And we will hear from Aziz Sancar in 30 minutes or so, his great Nobel Prize, and I'm sure he will be able to convince you better than I am how powerful genetics are. In general, we do research to understand normal, healthy, and as well as disease biology. This lets us, I'll get back to this point, hopefully for early prevention, but if not early diagnosis, and if you still cannot prevent the formation of disease, identifying genetic targets, therapeutic targets, and using specific targets, what we call as precision medicine now, that President Obama has really emphasized and really led. We do biomedical investigation to understand these normal and healthy diseases, but again, to really use these targets that we have identified. And if you think about genetics, and if you think about the last 2 decades or more, of how we identified these targets, these really came from rare disorders, looking at Mendelian disorders, because these are the pure forms of what happens of the human, if you will, gene, most of them gene knockout models. And then more recently, these approaches have been very powerful in understanding the mutations only also at the tissue level, looking at cancer, identifying targets in cancer, developing specific medications that target these mutations, and hopefully opening the door for effectively curing cancer. I'll show you at the end of my talk how for the most malignant brain cancer, glioblastoma, this silver bullet approach does not work. But especially in liquid cancer, in hematological malignancies, there's a big role for all of these medications, as well as in some solid tumors like lung cancer right now. In my lab, as Gökhan mentioned, we focus on vascular diseases. I'm not going to talk about that. I'm going to briefly mention some disorders of human brain development. Unfortunately, these are children born to consanguineous marriages, mainly in this collaboration is with over 21 universities now in Turkey, as well as I'll show you 2 small stories about brain tumors. What enabled us to unlock all these doors and make these discoveries was a technology that was developed at Yale and University of Washington in 2009, so-called exome sequencing. And the idea is very simple. If you look at these rare Mendelian disorders and look where the disease-causing mutations are, 80 to 90% of them are within the protein coding regions of the genome. The haploid genome is 3 billion bases, and of that, 1% encode for the proteins, and 90% of the mutations with large effect reside in there. So we said, why don't we just focus on that? And then why don't we do basically sequencing of all protein coding regions of the genome, so-called exome, and the exome approach developed. The initial cost of this was over $10,000. It's around $400 now. The idea is very simple. Take the genome, shear it down, use liquid hybridization to enrich for protein coding regions of the genome, and then after that, using bioinformatic analyses, just find the disease-causing variants. And this is now, of course, applicable not only for discovery of disease genes, but also identification of therapeutic targets, which I'll try to show, as well as making clinical diagnosis. This was a case, unfortunately, of course, consanguineous marriages. This is from Hatay. 2 kids affected, first cousins, marriages, both of them. And here's a control MRI. You can see, for example, if you look at this axial image, how the brain is really is nicely formed. Here it's much smaller, so microcephaly, which became unfortunately a common name now. But this also has what we call as Lissencephaly, which is the convolutions of the brain was developed to give us more areas to compute, and this is all smooth. So it's not only small, but also the surface is very smooth. To make a long story short, this was the first application for disease gene discovery of exome technology. Kaya and Ali in the lab took this, and other families applied exome sequencing technology and identified in these mutations so-called in WDR62, a sort of a novel gene. There was only one in vitro publication on this gene. And this showed the power of this technology. And this was recognized by Science magazine as one of the top 10 breakthroughs in science in 2010. And they used one of our patients. This is another patient, actually, this is from ARA. That is unfortunate, but they, in the editorial, they said that exome sequencing technology will change the way we approach the genetic diseases and how we identify disease-causing variants, especially at germline. They were correct, but as I'll mention at the end of towards the end of my talk, what has also had probably even more impact of exome technology has been in cancer. Since then, we have sequenced now over 1,000 children, 500 of them or so are with these really severe structural brain malformations. I won't go into those publications, except I will say you that as I was going to middle school and high school in Turkey, we called Anatolia the melting pot. And then it turns out that I think genetically that's also true. This is the genetic map of Turkey from over 1,000 patients. And if you look at it, this is sort of where Anatolia is, the red color. And really the population in Turkey is from 3 different genetic vectors. One that goes to Europe, the ones with the sort of the blue eyes, and then the one that goes to Caucasia, and sort of the more to the east, and the one that goes to the Arabic Peninsula and to south. But the majority of the population, at least currently, is really a mix of these 3 vectors to one degree or another. So as I mentioned, use of this technology for disease gene discovery has really been very powerful, leading to these new biological insights, leading to clinical advances, identifying clinical targets, identifying biomarkers and prevention, hopefully. But more importantly, as Gökhan said, as a practicing physician, for me, the important question is how do I apply it to my daily practice? What can I do about these? So the concept of personalized medicine was born, and I'll try to convince you that this is a reality today, that we exome sequence actually every brain tumor that is resected at Yale, and trying to give precision medicine to each patient. Of course, therapeutic targets are not identified in every individual cancer, but in some of them you can really make a difference, and I'll show you one specific example. Thinking about brain tumors, various types, or at least I should say the ones that originate in the intracranial cavity, and looking at the age and prevalence, we really focused on the most common ones, gliomas, which are the most common malignant cancerous brain tumors, and meningiomas that are the most common benign tumors that really are by far mostly in the population, to the extent that these are also driven by estrogen. And in autopsy studies, up to 3% of women actually have a small meningioma, not the one that has clinically important, but they're extremely common. Talking about next generation genomic technologies, I mentioned the exome sequencing technology, which really remains the workhorse, and that's what we do. But when we take a brain tumor, for example, we not only do exome sequencing, we also used to do whole genome genotyping, looking at large-scale chromosomal abnormalities. You really don't need to do that anymore. Whole genome sequencing will become the standard. If you think about that, the first human genome sequencing, 1 million base pairs costs around $100,000. It's down to around a couple cents. So the development of technology has been amazing. There's still significant problems with analysis of the whole genome sequencing data. We'll talk about Turkish Genome project, but the reason for that is that the need for the computational power is exponentially higher to analyze these. And you approximately generate 2 trillion data points after one genome sequence. Of course, methylation with the epigenetic modulation of the gene expression, as well as chromatin immunoprecipitation sequencing, looking at these epigenetic changes, are really important, as well as the recent introduction of single-cell technologies looking at these individual cells in cancer is really exciting. So given all of those, I'll show you two, as I said, I'll give you two stories. One of them is looking at these meningiomas which originate from the meninges, the membranes surrounding the brain. These are benign tumors. They rarely become cancerous. The problem is that they can originate anywhere in the brain, or I should say along the meninges, either in skull base and infiltrating structures like the vascular structures or important nerves. So Victoria and Zeynep took 50 of these tumors and applied exome sequencing technology, and the results are shown in a single slide here in a graphic format. Here, these are the expected number of mutations. These are the mutations that they observed. And they observed that the 50% or so meningiomas had this mutation in the tumor suppressor gene called NF2, which was a good validation of the study because this was previously reported. They have seen mutations in 2 genes that were reported in cancer before, including AKT1, which is a part of this signaling called PI3 kinase, which is important for survival of cells, as well as this gene called SMO, which is involved in this pathway called hedgehog, which is important for embryonic development, especially of the face. But there were these 2 other genes, TRAF7 and KLF4. This was published in 2013. Other publications confirmed our findings, but most beautifully, and there's a rare finding, there was a really clear distinction between these tumors that harbored these mutations, such that they formed really 3 distinct classes. Ones with the NF2 loss, ones with the TRAF7 mutations, and ones with the SMO mutation. And with TRAF7 mutant meningiomas always existed with one of these 2 genes, mutations in one of these 2 genes. As I mentioned, AKT1 mutations have previously been reported, but KLF4 was a fascinating gene to us because this is one of the 4 transcription factors, so-called Yamanaka. This is 2012 Nobel Prize. When you basically take any cell, a skin cell, and you might have heard this in the news, and put these 4 transcription factors, you can actually induce formation of stem cells. So these 4 transcription factors are sufficient. And the mutation that we identified was in many different meningioma samples, but always affected the same part of the gene, of the protein, and at the DNA binding domain. This suggested that it actually gave it a new function, which I won't go into details. But going back to these, and this is probably why it's important that the physician scientists, endangered species even in the U.S. continue, because normally you would stop there, but we ask, is there any clinical correlations? Can we really, based on these beautiful distinction between these groups, do these have any clinical meaning? And the answer was yes. Victoria and Jennifer went back and looked at the origin of these meningiomas along the skull and the brain. Turns out there was a really clear gradient, such that, for example, NF2 tumors were at the back of the head, whereas the other ones were in the front. And if you just pay attention to this mutation that is really restricted right here in adults, all the meningiomas that originate from here, which the smell nerves come, so olfactory nerves come, were localized there. And these are, again, mutations that happen at the tumor or the cancer level. When you look at the mutations affecting this gene or the pathway in newborn, in germline, they have a very similar phenotype. This was reported in the 1990s. These babies, when they have germline mutations in hedgehog or SMO, the brain fails to split into 2, so there's a uniform brain, and they have a single eye, cyclops. You might have heard the myth cyclops. That's due to mutations of these. The same gene mutations in adults causes meningiomas exactly at the same location, which I find quite fascinating. So as I mentioned, these were confirmed by other publications. But most importantly, looking at the clinical cancer advances in 2013, they thought that these analyses really opened the door for personalized treatments, because when you think about these mutations, of course, luckily there are limited ways that nature can create cancer and keeps using the same pathways over again. The SMO mutations, for example, are important for basal cell skin carcinoma, and there are actually FDA-approved medications for those. So a trial is ongoing for meningiomas that localize exactly the same region. The last story in the couple slides that I'll say is this problem that fascinated me for a long time is of gliomas. I should say 20% of gliomas start like this, looking like this on MRI. Very well localized, benign tumors. But ultimately, 100%, and there's something rarely 100% in medicine, but at least all the ones that everybody have followed, become this really malignant cancer called glioblastoma, They can be dormant like this for decades, but then they kill the patient within a year. So we studied the molecular mechanisms of that. And turns out that it's quite complex. It's not a progression like colon cancer that the cancers just keep accumulating mutations. If you just focus on this panel, in blue are the ones that mutations that they acquire, and in gray are the ones that they lose. Meaning that this is actually a dynamic nonlinear clonal progression. These tumors acquire mutations, lose mutations, and ultimately become malignant like this and unfortunately kill the patient. And if you just focus on this panel here, this is the benign tumor, this is the malignant tumor, this is the clonality, meaning that what percent of tumor cells has a specific mutation. In this benign tumor, all these mutations that were in 100% of tumor are all gone as the tumor progressed. So a lot of mutations are lost, a lot of mutations are gained, and ultimately, it's a busy slide. You don't need to pay attention to this one, but a lot of different pathways are activated or lost, leading to formation of this cancer. But all these changes, and this study was published just 2 months ago, allows us to come up with specific treatments. Because most of these changes are epigenetic. We use a specific kind of drug called bromodomain inhibitors, affecting those epigenetic changes. And for the first time, these drugs were used in glioblastoma and really obtained a therapeutic effect. As I said, this is now real time. Every Monday, we meet along the tumor board, discuss these findings, come up with treatments. And then this leads to formation of this precision medicine board. Not only we focus on discoveries, but also targeted treatments. As I said, there's no silver bullet. And unfortunately, this is one of our biggest success stories. A patient who unfortunately passed away recently was diagnosed with glioblastoma in 2010, life expectancy of 12 months, survived 5 years, recurred twice. Every time we have given specific treatment because the cancer kept changing, we were able to keep her alive, but not, unfortunately, cure her. So the last slide, Gökhan, is the transfer of technology. So we have formed a collaboration with TUSEP and the Health Ministry. The idea is that we have this lab, the genome analysis lab at Yale, not only, again, the infrastructure with the sequencing machines, but also the computer infrastructure. We're hoping to establish this in Turkey. This is in collaboration not only with TUSEP and Health Ministry, but also with academia. We are going to train over 40 individuals from the technician to the director to the bioinformaticians at Yale over the next 3 years. And we hope that this will be the first phase of the Turkish genome project, which Fatih Nocu tells me that they're going to sequence 100,000 Turkish genomes. So that's a very exciting, very great project. So thank you very much.
So we can take a few questions while they're setting the computer up. Quick ones, please. Yes.
Wonderful talk. Thank you so much, Murat. I have a question about the tumor aspect that you have shown and neurodegeneration. I know your lab works on neurodegeneration as well. Do you see any common or unique pathways? Do you see, for example, patients who have either cancer or cancer in their family, and they have a different perspective or a reflection of their genome? How do you see this cancer neurodegeneration affects your results or findings?
I think there, again, given the fact that, and you'll probably be able to answer that question better than I do, but there are certain pathways that cancers kept activating. In the, what I haven't shown is that the ultimate, all those pathways converge in malignant glioblastomas into locking these glioma cells into a stem cell-like state, de-differentiate them, and kept them in that state, locked them in that state, and inducing proliferation, continuous proliferation. So they're just jailed in this area where they're embryonic-like stem cells, and they keep proliferating. And I think some of the neurodegenerative pathways are going to be important in some of that, and they will be probably specific for the CNS, the central nervous system tumors, but happy to discuss further.
Please.
Hi, that was a very interesting talk. I'm just curious about your...
You should see the other 100 slides.
I was curious about your thoughts of identifying highly heritable diseases that have a lot of genes with small effects that play a role in that, and how you would use genomics for that.
Thanks for bringing that up. I think that's the holy grail that we're just getting into, and we actually discussed this as a part of the Turkish Genome Project, because one of the huge advantages of Turkey is the electronic medical records. U.S. Has not obtained, and this is a little, I'll just give you a short answer, we can discuss further, but has made two mistakes. Number one, they did not allow prospective calling of patients, because as you sequence, you find a variant that you know that will be important diabetes, you want to be able to call the patient back. They did not do that. UK did that. They're sequencing 200,000 people. The second thing is that the electronic medical records are not uniform here, so you cannot really get that. But if you think about it, when you try to find a disease gene for a rare Mendelian disorder, the matrix is 30 million variants with those spots, and you look at 100 patients. So your matrix is 100 by 30 million. Now you're going to be looking at 3 billion by a matrix of probably 10,000 patients. It's a very different scale, but that's exactly where we need to go as a field, where the field is going.
Awesome. From
Brief,
Northwestern. It's very interesting to see the activation of a mutation in a very specific place. So it's very amazing to see that. Are these mutations kind of coming from the germline, or are they being acquired? And how fast are these gliomas, glial cells proliferate to acquire such a mutation in that environment to become kind of malignant?
If I understand the first part of the question, you're asking about recurrent mutations, why it's affecting the same spot on the protein?
I thought it was very interesting to see that the mutation being active only in histologically on the same spot, given in association with a specific disease. That's very amazing. But my question, whether it is coming in germline or it is being acquired, and if it is coming with germline, why it is being activated?
He's going to kill me, sorry. Right? But let's discuss more. But it usually is somatic, meaning that acquired at the tissue state. It usually means that if there's a specific mutation, activates the function of the gene or gives a new function of the gene. It's very easy to lose the function of gene, like BRCA, breast cancer gene is a good example, tumor suppressor. You can mutate it anywhere in the gene. But to activate it, you only have 1 or 2 shots. So it's my pleasure to introduce Professor Hotamishligil, and I had to take notes because it's long. But he's currently the Simmons Professor of Genetics and Metabolism and has been the longtime chair of the Genetics and Complex Diseases at the School of Public Health and at Harvard. And recently he became the director of Sabri Center for Metabolic Research. That we need to understand how he did that. But that's another story. So he specifically focuses on obesity, diabetes, and heart disease. And I'm really honored to be his friend. And he created this field called immunometabolism, where he specifically looked at the immunological mechanisms for formation of diabetes, which was a really new field that he pioneered. He quotes 180 papers in his CV with over 40,000 citations. It is true when I say that he has too many awards to list, but what's relevant to us from Turkey is that he's an elected member of TUBA. He got the TÜBİTAK Science Award, Vehbi Koç Foundation, and many others. So look forward to your talk.
Thank you very much.